This is what people looked like in Western Europe and Britain 10,000 years ago, not what we would have expected.
1, 2, 3, come on!
And I'm holding here this wonderful reconstructed head of Cheddar Man. And I've been studying the skeleton of Cheddar Man for some 40 years or so. And now with DNA technology, we've got the whole genome of Cheddar Man. And here we've got a scientific reconstruction of what he looked like. We've got his hair form, his skin, his blue eyes, and that dark skin colour, that combination really striking. And getting out of here information about his biology, the fact that, for example, he couldn't digest milk.
For several years, the media have highlighted studies claiming that ancient Europeans were black and that white skin only appeared in Europe about 8,000 years ago. The most well-known of these studies is the case of the Cheddar Man, a skeleton found in England that is believed to have lived around 10,000 years ago. It has been globally presented as evidence that Europeans were black during prehistory. In this video, we will demonstrate why this study is completely false and why its genetic results have been heavily manipulated. Firstly, a significant point is that the mitochondrial DNA of Cheddar Man belongs to haplogroup U5b1. While haplogroups do not directly determine skin pigmentation, they remain an important indicator, allowing us to compare him with modern populations. U5b1 is a haplogroup of European origin. Over 80% of the mitochondrial DNA from European Mesolithic individuals belongs to various subclades of U5. Burial sites with haplogroup U5b1 have been found in England, Germany, Switzerland, and France. Moreover, modern Western Europeans show a significantly higher frequency of U5b than U5er, with Basques almost exclusively carrying U5b lineages. Approximately 11% of modern Europeans belong to haplogroup U5. The U5 haplogroup and its subclades U5er and U5b have their strongest concentrations in Nordic populations, particularly among Finns and Estonians, but are also widely distributed across Europe. The main argument used to claim that Cheddar Man had black skin is the absence of the SLC24 and SLC45, A2 genes, which are associated with white skin. This reasoning is also frequently cited in other articles, arguing that white skin only appeared in Europe 8,000 years ago. This leads us to an important question. Do we really need the SLC24A5 and SLC45 to have white skin? Ancient Basques, Hungarians, Finns, Siberians, and even the Ainu, for instance, do not carry the SLC24 and SLC45 genes, yet they are not black. They clearly have white skin. So, where does this confusion come from? It is true that today, the majority of white populations have inherited the SLC genes due to interactions and mixing among Europeans over time. This does not mean that these genes are necessary for white skin. Originally, one European population among many possessed this specific gene, and through intermixing managed to pass it on to most European peoples, except for a few groups that were more isolated, such as the Basques or the Hungarians. In the past, there were many European populations that did not have these genes, but were still white through other genetic mechanisms, much like the Basques today. A simple analogy explains this misunderstanding. Apple is a fruit, but not all fruits are apples. Just because the majority of white populations now carry the SLC genes, does not mean that these genes are necessary for being white. This is a logical fallacy. The reality is that the SLC genes became dominant and replaced other genes that already had similar characteristics. This explains why we find ancient European populations that did not have these genes, but this does not mean they were black. For instance, the Japanese also do not have the SLC genes. Are they black too? Absolutely not. For years, geneticists have been trying to uncover the origins of physical traits through genes, but when we delve deeply into the genetic mechanisms, such as those of skin pigmentation, the situation is far more complex than the SLC24 and SLC45 genes might suggest. The evolutionary history of humanity has led to considerable variability in the mechanisms behind skin pigmentation. In essence, there is a wide range of genes that can contribute to certain traits, which means we cannot judge an individual's appearance based solely on the presence or absence of a few specific genes. Here's what Brenna Henn said, a geneticist and researcher in the Department of Anthropology and Genome Sciences at the University of California. The known pigmentation genes, discovered primarily in East Asian and European populations, do not explain the variation in skin pigmentation. The idea that there are only about 15 genes underlying skin pigmentation is not correct. It now seems likely that many other genes influence skin color. We do not know how. If we are still learning the connection between genes and skin within populations, we cannot yet predict the skin color of prehistoric humans, says Brenna Henn. This highlights the fact that to determine someone's racial identity, it is essential to use every tool at our disposal, not just limit ourselves fallaciously to a few genes. Instead, we must analyze as much data as possible, including haplogroups, entire DNA sequences, skull shape, skeletal structure, or even hair color if dealing with a mummy. The theory of black Europeans becomes even more absurd when we look at Scandinavia, where a study found 7 hunter-gatherers in Sweden, dating back 7,700 years, who indeed carried both light-skinned genetic variants, SLC24 and SLC45. They even had the HERC2 OCA2 gene associated with blue eyes and blonde hair. These populations, as they migrated southward, passed the SLC genes to other Europeans. However, as explained earlier, while these genes are part of those associated with such traits, they are by no means a necessity for having these features. Now we will demonstrate why the Cheddar Man study is, in fact, a massive fraud. Here is the software researchers used to decode Cheddar Man's DNA. This tool was specially developed for this occasion by Susan Walsh of Indiana University. It is a model that attempts to predict a person's eye, hair, and skin pigmentation based solely on their DNA. To test it, Miss Walsh and her colleagues collected genetic data from over 1,400 individuals, mainly from Europe and the United States, but also from Africa and Papua New Guinea. When the details of Cheddar Man are fed into the software, here's what you get. The first thing you should notice is the lack of precision in the results and some clear contradictions. The individual is predicted to have either black skin or brown skin, but zero chance of having dark brown skin. That's really weird. Then, the software states that he would have light-colored hair, despite having a 70% probability of brown hair, over 20% chance of black hair, and very little likelihood of being blonde or red-haired. As for the eyes, it predicts a 20% chance of brown eyes and a 50% chance of blue eyes. This is far from precise. So, did he have blue eyes or brown eyes in the end? The problem with Cheddar Man is that we cannot compare the study's results with how he actually looked in life, because he is dead a long time ago. However, to assess whether the software created by Indiana University is reliable, we can input our own genetic data and see if the description generated matches our actual appearance. An internet user tried the tool using their fully sequenced DNA test provided by a highly reputable laboratory. The user is entirely British, with very fair, milky white skin typical of the English blondish hair, and very dark brown eyes. Here is what we get. According to the same software used for Cheddar Man, it predicts that this individual does not have white skin, but rather brown skin with a 90% probability. It also claims they have a 90% chance of blue eyes and only a 0.01 points of having brown eyes, despite the fact that this person has very dark brown eyes. These results clearly show that the software is unreliable even when analyzing recent fully sequenced DNA. This is further compounded by the degraded state of Cheddar Man's DNA, as noted by Brenna Henn. We cannot place such trust in DNA analysis. Cheddar Man's DNA has degraded over the past 10,000 years. In fact, his DNA ultimately holds no value, especially considering that even with a complete DNA sequence, errors already occur. The model operates using a percentage-based system. The less DNA you have, the more influence the incomplete genes of the remaining DNA will have in the software. This artificially inflates certain traits in the absence of other data. Additionally, as we will see later in the video, scientists have still not managed to precisely identify which genes determine skin color. Beyond this initial issue, the tool in question uses data from only 1,423 individual samples, which is far too few. As a result, it relies on just 36 SNP's single nucleotide polymorphisms to determine Cheddar Man's skin color. This is insignificant on the scale of human genetics. Currently, over 120 million SNPs are known worldwide, and the lack of diverse data in the software renders it completely inaccurate. We decided to use other software to analyze our internet users' DNA and compare the results with those of Indiana University's tool. Here's what the model from 23andMe, known to be much more robust as it uses between 700,000 and 1 million SNPs, reported. This is far beyond the 36 SNPs used by Indiana's HighRiskPlex software. First, we observed a 40% probability of very fair skin, whereas the software used for Cheddar Man claimed our internet user had a 90% chance of brown skin. The difference between the two models for the same DNA is enormous. It's simply not the same result. And yet, even though the software is accurate regarding eye and skin color, it remains terribly unreliable in other aspects, as shown by the results for hair. It assigns a 50% probability of having dark hair and less than 2% for blonde hair, even though our internet user is blonde. These genetic models are essentially like playing Russian roulette. Here's what Brenna Henn said about her colleague in the New Scientist article. Even Dr. Walsh herself insists that the study does not conclusively demonstrate that Cheddar Man had dark to black skin. So why conduct misleading studies that the media then amplifies? It's also worth noting that SNPs such as those associated with eye color like RS129 are often misinterpreted. While it was initially thought that SNPs functioned identically across all humans, the reality is that genetic and racial variability among human populations is so extensive that the same genes can have different effects depending on the individual or population. This is referred to as inter-individual variability. Consequently, similar genes may not act with the same intensity and can result in different traits depending on the individual. Some studies suggest that the white skin of Europeans developed due to migrations of populations from the Middle East and the Caucasus about 8,000 years ago. However, detecting a gene in an individual from that time and place does not necessarily prove that this trait originated there and then. This is yet another example of a total paralogism. The theory that light pigmentation in Europeans is a product of specific populations, whether the Yamnaya people or Anatolian farmers from 8,000 years ago, is entirely unfounded. As we've shown, the presence of a few specific genes cannot reliably indicate an individual's physical appearance. There are far too many genes involved, many of which remain unknown to us. Additionally, mutations responsible for traits like skin pigmentation or others can arise independently and in isolation within different human populations. This makes it impossible to establish a straightforward linear timeline for such developments. For instance, SMP-RS 142 highlights this complexity. These theories are so absurd that the primary gene SLC24, supposedly responsible for white skin in Europeans around 8,000 years ago, has actually been present for millennia in African populations such as the Khoisan. These groups evolved in isolation in Africa, in warm environments, further disproving the idea of a singular linear evolution of white skin. This demonstrates the variability in how the same gene can act within two different races, and how it can arise in various ways within geographically separated groups. This phenomenon is the principle of evolutionary convergence. Different species can develop similar genes over the course of their evolution without ever having come into contact with one another. The case of the SLC24 gene, found both in Nordic populations and black populations, shows that it is not the key factor responsible for white skin. This disproves any link between white skin and issues such as vitamin D deficiency, lack of sunlight, or climate, as this gene is present both in Europe and Africa. In Central Africa, for instance, there are subtropical and mountainous populations with limited sun exposure, equivalent to levels found in Europe and North America, yet they have retained dark skin. What Wikipedia and certain studies tell you about white skin is false. They deliberately want you to believe that white skin has a recent history, that it is unrelated to your past or your ancestors. Your identity is made to seem optional, superficial. Even the media and some scientists try to implant the idea in your mind that blue eyes aren't truly blue, offering various abstract justifications. They claim blue eyes are actually brown. According to these narratives, you're all just brown or black, and it's systemic racism that has made you think otherwise. These same studies claim that blue eyes are recent and the result of a random mutation in a single individual a few millennia ago. It's always the same idea. They want to deceive you. The claim that all people with blue eyes share a single ancestor from the Black Sea region 10,000 years ago is a fairy tale, a story made to lull you into complacency. The same mutation occurs repeatedly in various locations and eras. As we've discussed in the video, blue eyes are not dependent on a single gene, and attempts to pinpoint which specific genes produce certain traits have largely failed. The study in question analyzed 800 individuals from Europe to the Caucasus. 800 people from modern populations were used to try to explain the origin of blue eyes throughout the entire history of humanity. Claiming that all individuals with blue eyes descend from a single ancestor who lived near the Black Sea 10,000 years ago is a misrepresentation of scientific data. The idea that blue eyes stem from a single mutation in a specific region relies on the study of a mutation in the HERG2 gene, which regulates the expression of the OCTA2 gene, crucial for eye pigmentation. However, this does not mean that a single individual was responsible for it. nor that it was confined to a specific region like the Black Sea. Mutations can arise independently in different populations. Blue eye coloration results from a genetic spectrum, not a single mutation. Furthermore, certain populations in Central or Northern Asia also exhibit light-colored eyes, but do not share this specific HERC2 mutation. This suggests they inherited blue eyes from another now extinct white population that did not carry the HERC2 gene, just as the Basques inherited white skin without the SLC genes. These recessive light-colored genes are clearly not characteristic of Asia, but were inherited from various European influences. The notion that this mutation dates back 10,000 years is also questionable. Ancient human remains from Motala, Sweden, dating to around 7,700 years ago, show individuals with blue eyes. However, this does not mean that the mutation originated at that time. It is far older. Claiming otherwise is as absurd as saying blue eyes first appeared in 2025 just because you meet someone with blue eyes in 2025. It's utterly nonsensical. The distant ancestors of white populations were already white. We didn't suddenly, as if by magic, gain blue eyes and white skin 8,000 years before our era. In fact, genes structure and organize themselves within individuals primarily based on the entirety of their organism. A gene on its own means very little because it has a wide variability of effects within the organism. A gene does not allow us to determine the biological or racial identity of an individual. This helps us understand that race encompasses thousands of factors beyond the simple presence or absence of a few genes. Races are complex biological entities that must be studied in their entirety, and the history of ancient populations reveals far greater diversity than what we are often led to believe. Here's what some specialists have to say on the matter. The hope placed in DNA sequencing as a singular answer to the question of origins rests on an illusion. The information DNA is supposed to reveal will only become true knowledge when DNA is considered in relation to the entirety of the organism in question. Knowing the sequence of a gene alone contributes very little. The problem is that there is simply no such thing as a representative sequence of the human genome, or any genome for that matter. The number of possible variations in the DNA sequence is far too vast in a natural population. Genes do not have a concrete existence. At least in the cells of higher organisms, there are only parts of genes that cells can use when they want to build a protein. A gene is in no way a molecule that exists in a cell. Genes are not, they only become. Regardless of how one defines a gene, it is clear that single cause and one-dimensional models, as required by the dogma of molecular biology, no longer hold. Despite this, the ideal persists of wanting to find mechanisms that form the basis of hereditary processes. However, a gene is not defined solely by its molecular structure, but, above all, by its relationship to its effects on the organism. Thus, genetics should not be reduced to isolated pieces of data, but must align with a comprehensive study of an individual's or a racial group's organism, taking into account other essential factors. Understanding the world and the way things function should never be limited to a narrow analysis of a few genes. Similarly, we must avoid any form of reductionism and instead strive for a holistic and global approach to things. This video serves as a good example to illustrate how the absence of a few genes, selectively and fallaciously highlighted by certain researchers, can lead to a host of lies about the existence of so-called black ancestors in Europe. This observation exposes the manipulation and intellectual dishonesty present in certain scientific circles. These revelations offer a fresh perspective on our past and encourage us to stop blindly trusting what the media or the academic world tells us, merely based on the pretext of academic qualifications. The scientific world is complex, and opposing theories are an integral part of science.